Oi, pessoal. Já vi que estão entrando, então tá tudo certo. Já tem o link para a chamada ali. Podem ir preenchendo. A nossa convidada já chegou. Eu vou pedir para ela aguardar até o horário para dar tempo de todo mundo entrar. Tá. Esquece o link da chamada. Eu vou arrumar e daqui a pouco eu mando de novo. Bom, peraí que é muita tela aqui. Ok, bom dia, good morning. Como a nossa convidada é internacional, eu vou falar em inglês. Then, since our guest is an international guest from Canada, I'll start to speak in English. I guess you are seeing us. Then, good morning, Sarah. Thank you for being here today with us and share your knowledge in this webinar. I will ask you to put your presentation because I think we can start in less than 1 minute.
Can you see it?
Yes, it's perfect.
Okay, excellent. So in case, because right now I don't see anything except this,
Yes.
the presentation. So in case there's something off with my audio or things like that, please interrupt me so that I'm aware.
Yes, no problem. Well, it's our time, 10 a.m. here. I guess it's 3 p.m. Where you are. Then we can start. Good morning to everyone. Our guest today is Professor Sarah Hedtrich from University of British Columbia. And thank you, Sarah, for share your knowledge and be available to be here with us today. I will turn off my camera and my microphone, but I am here seeing and listening to you. Then in any case, you can just call me. So good webinar for us.
Great. Thank you so much for this nice introduction and for inviting me here. It's really my pleasure to present in your webinar today. And over the next 40 to 45 minutes, I will talk a little bit about our novel approaches to tackle inflammatory and genetic diseases of human epithelia, which is really the focus of my research lab. And so the overarching theme is where my lab is striving to understand and tackle inflammatory and genetic diseases, especially focusing on the skin and the lung. And under this overarching topic, we have defined 3 main research areas in that we are working. One is bioengineering, which is basically motivated by the fact that for most of the diseases we are interested in, no good or no suitable animal models do exist, and therefore we are bioengineering our own human-based disease models that we then leverage to study disease-related mechanisms, and that we also use for the development and testing of next-generation therapies. And so this whole focus on the human-based disease models that we are doing is really motivated by the problems in the drug development process today. And I'm pretty sure that most of you are aware of that. The drug development process today is often referred to as a death valley. And the reason for that is that we are currently see unprecedented high investments into drug development, and they increased over the past decades tremendously, while at the same time we still have a very high failure rate when it comes to drug development. So the average drug attrition rate across all indications is 90%. In some indications, we are looking at close to 100%. And this is, for example, the case when talking about neurological disorders or things like Alzheimer's disease. But this drug attrition rate, which means these are the percentage of drugs that never make it to the market or that are never being approved, this actually hasn't changed over the past decades, although we have this significant boost in investments in the drug development process. And so there are several reasons that account for that, but one central aspect really seems to be the models that we are using. And as you probably are all aware, these are mostly animal models, and there are clear species-related differences between animal models and humans, and very often animals have different diseases than humans have. And so what we are often trying to do is to induce a disease phenotype in an animal that somehow emulates the disease or the human disease. And then we are expecting that this disease phenotype that we induce in an animal is basically predicting the human response to treatments, or that's basically in line with the human pathophysiology. And very often that's actually not true, which means that the results from animal models towards the human situation is often cannot be easily transferred. And in addition to those interspecies-related differences, there's another good reason why one should focus on human-based models. And this is that we have definitely entered an era of modern drugs. And modern drugs do require human models. And just to corroborate that statement, it's basically almost half of the drugs that are currently under development. These are so-called biologics, so things like antibodies. At the same time, we also see a significant increase in genetic drugs. So in 2018, the very first siRNA-based drug has been approved. We have seen the success stories of the COVID-19 mRNA vaccines. And currently, there are first clinical trials ongoing that leverage gene editing tools like base editors or CRISPR-Cas9 as a new therapeutic approach. And all the majority of those biologics like antibodies, but also gene therapies, they are highly specific. Which means that they are designed to recognize a specific human target. And the thing is that very often those targets are not necessarily preserved in animals, which means that a certain biological molecule, as we know it in a human, exists in an animal, but it has a completely different structure or a different amino acid sequence or things like that. And so if you want to translate or directly translate the findings from animals to human, you have a problem, because you basically have to go back and redesign the system, because here we are really working with very specific approaches. And so it would be definitely easier to work with human-based models early on, because then we wouldn't have those issues of interspecies translatability of the findings. And so because of that, that's really the motivation for my lab to develop human-based models and to use them to study disease-related mechanism, but also for developing new treatment options. And so over the next few minutes, I will present you a few projects that my lab is working on, which are located in one or the other focus area of my lab. And I will start with a story that is really located at the interface between the bioengineering and the molecular medicine approach. And this is a story that builds around atopic dermatitis. So as mentioned at the beginning, my lab is very much interested in inflammatory skin diseases, and the inflammatory skin disease we are most interested in is atopic dermatitis, which really is the most common inflammatory skin disease worldwide. It currently affects up to 25% of children and up to 7% of adults. It is a chronic disease, a relapsing disease, which is driven by a Th2 immune response. And we also know that it's multifactorial and a very heterogenic phenotype. And what we also know is that patients suffering from atopic dermatitis also have a highly impaired quality of life, and that you can probably appreciate that when you have a kid with such a severe atopic dermatitis, which is often associated with a lot of itching, that it's really easy to guess that this baby doesn't sleep well because it's itching all the time, and the parents won't sleep either. So this is really a vicious cycle, and there are some studies that show that people suffering from moderate to severe atopic dermatitis experience the same quality of life like people suffering from cystic fibrosis, which eventually is a fatal disease at some point. The interesting thing about atopic dermatitis, however, is that very often this inflammation can progress to other epithelia in the human body. And this progression is called the atopic march. And the interesting thing about that is that it almost always manifests first in the skin shortly after being born. And then especially kids suffering from moderate to severe disease activity, a large percentage then also develops food allergies when they turn 1 or 2. Some of them then also develop allergic asthma around an age of 5 to 8. And some of them in early adulthood also develop allergic rhinitis or hay fever. So there's clearly a progression from skin to the other epithelia in the human body, but we don't really understand why and how this is happening. And part of the reason is that we, again, lack suitable models, because very often in dermatological diseases, mouse models or rodents in general are being used. But the thing is that mice, for example, do not spontaneously develop eczema or atopic dermatitis. And it's also very difficult to induce something which comes close to the progression of atopic diseases within the mice. So basically, we are lacking suitable models to study that. And so what my lab has been doing over the past 10 years is to generate skin disease models, and this is the lower part here, so 3-dimensional models that emulate characteristics of atopic dermatitis. And we do this by knocking out a disease-relevant gene, which is filaggrin. Then we add important disease-relevant cytokines, such as interleukin-4 and 13 to that, and we know that by doing so, we can generate a model that emulates key characteristics of atopic dermatitis in vitro. And so my lab was particularly interested in how the skin communicates with the other epithelia in the human body. And so what we do is we started putting different tissue models together in so-called multi-organ chip setups. So what you do with this, and this is a schematic drawing of that, is that you have a microfluidic chip-like setup where you can incorporate different bioengineered tissue models, like in our case, for example, the skin disease model and a lung model. And then you can attach that to a pump or connect that to a pump. And this pump can really emulate the systemic circulation, the human blood flow. You can also add immune cells to the circuit. And by doing so, you are trying to basically emulate the parameters within the human body. And so what we do in this setup is we developed a so-called ex vivo drug discovery platform for atopic diseases. And this is what those chips look like. And here specifically, we're implementing the skin disease model in one of those big chamber, another one, the lung model. We are also adding immune cells, which are relevant for atopic dermatitis, into this chip. And another student of mine is currently working on bioengineering a lymph node-like structure, which eventually will be integrated here as well, because there's a strong immunological component to atopic dermatitis. And so by leveraging those setups, what we really wanted to understand is how does atopic skin communicate inflammation to the lung? So basically, we are postulating that there's something like a skin-lung crosstalk, and that's what we wanted to address. And so this is really the question, how does skin communicate inflammation to the lung? And we started off by a simple co-cultivation between inflammatory skin disease models and bronchial epithelial models. And just by looking at the histological staining, the effects become quite apparent. So I will, this is a busy slide, but I will walk you through. So what you can see here in the top panel, this is basically the histological section of a normal bronchial epithelial model, so basically lung tissue. And this is a normal H&E staining. What you see here is the bronchial epithelium, the erected cells. And here on top of that, you see the cilia, which are, by the way, beating in a physiological velocity. We know that. And what you see here on top, here it's better visible in bluish. And this is actually the mucus. So those lung models produce mucus, which, you know, is also happening in the physiological lung. And after we co-cultivated this healthy bronchial epithelial model with our atopic dermatitis-like model, this is what we observed, which is, for one, we see a hyperplasia of mucus-producing cells. So here it becomes even more visible, so they become very, very big. And what we also see is the upregulation of pro-inflammatory markers, which are also very often upregulated in asthma patients. And these are things like POD2, uroglobin, alpha-SMA, TSLP, and we also see a downregulation of some barrier proteins in the bronchial epithelial model. So obviously, this co-cultivation alone triggers an inflammation in the lung model. And in the next step, what we wanted to know is what exactly triggers this inflammation. And so it seems to be something that the skin models secrete. And so what we did was we collected the culture medium of the skin models, and then we subjected them to electrophoresis, and then we identified some bands which are different between the disease models and the normal models. And then we analyzed those bands, we cut them basically out, and then we performed mass spec analysis in order to identify what proteins are present in those very specific bands. And what we found, which was quite intriguing, is that the compounds that we identified there were all had to a certain extent a connection to the so-called skin matrisome. And the skin matrisome is basically the entirety of the extracellular matrix of the skin, which is quite interesting, because what we found, things or factors that we found to be elevated are things like CD44, there's also thrombospondin, and there's fibronectin. And why we were excited about that is basically because it's known that changes in the extracellular matrix composition that this is closely connected with inflammation. And this is a paper that has been published before, and this is related to another epithelium, in fact, the intestinal epithelium. But here what they basically show is that the extracellular matrix undergoes a remodeling, and that this is really closely connected with the inflammation in inflammatory bowel disease. And so this, together with the results from the secretome analysis, basically got us interested. And so we started looking in more detail in the extracellular matrix of the models. And what we did was we collected fibroblasts from atopic dermatitis patients, and we cultivated them in a way where they produce their own extracellular matrix, and then we characterize this extracellular matrix. One thing was doing that by scanning electron microscopy. And so what you can see here on the left side is really the nicely aligned collagen bundles as we find them in the extracellular matrix that was produced by healthy fibroblasts. For comparison, this is the extracellular matrix of native human skin, and I hope you can appreciate that it looks more or less the same, although I should say that the bundles here look a little bit thicker than here, but the structural organization is very alike. And now look at that picture here. This is what the extracellular matrix looks like in fibroblasts, in atopic dermatitis patient fibroblasts. And I think it's quite obvious that they are not well aligned and that they are structurally somehow disorganized. And in addition to this structural disorganization, what we also found was that there is significantly less collagen in the extracellular matrix of atopic dermatitis patients, and that there is significantly less elastin in comparison to the healthy controls, which is a quite distinct difference. And so we went on and we wanted to know in more detail, how does this extracellular matrix produced from atopic dermatitis fibroblasts differ from normal fibroblasts? And so we conducted a proteomics analysis. And so we found all those red dots that you see here in this volcano plot. These are proteins that are differentially expressed in healthy ECM versus atopic ECM. And so those where you see the label next to the red dots, these are where this difference is most pronounced. And so I circled some that were of particular interest because, for example, what we found is that tennessine is obviously significantly more expressed in healthy extracellular matrix, and we know that tennessine binds fibronectin. And now when you think back, fibronectin was one of the compounds that we found as a secreted factor in our skin disease model. And so we think that if there's less tennessine in atopic ECM, there's a less binding partner for fibronectin, and therefore we find that in higher amounts in the culture media. What we also found was that in atopic extracellular matrix, things like the serine protease 1 was upregulated, and this is an enzyme which contributes to extracellular matrix remodeling. At the same time, we find STAT1 upregulated, which is important in fibroblasts when they are undergoing a pro-inflammatory state. And on the other hand, we found significantly less superoxide dismutase in atopic ECM, which is responsible for the deactivation of radicals, oxygen radicals, for example. So this all points towards a remodeling in extracellular matrix in atopic dermatitis. And so we complemented that by running some transcriptomic analysis with the fibroblasts, and that the data are basically well in line with what we found at the proteomic level. So the pathway analysis that we conducted really showed us that there's something going on with regard to extracellular matrix organization, degradation of extracellular matrix, collagen cross-linking. We also got hits like interleukin-4 and 13 signaling, VEGFR signaling, and this all points into the direction that obviously extracellular matrix or the fibroblasts in atopic dermatitis patients behave differently from their healthy counterparts, which is a very interesting finding. And so now, in order to see what this will mean in a like actual more complex setup, this is one of the rare occasions, really rare occasions, when we then proceeded and conducted an in vivo experiment in mice. Knowing or being aware of the limitations that may have, however, since we only can emulate a rather limited number of tissue interactions in our setups, we wanted to see if we can find some pro-inflammatory effects in a complex living organism. And so what we did was an in vivo experiment where we implanted pumps that were filled with some of the mediators that we identified as potentially interesting. And this pump was implanted on the back of the mice. And then we injected those mediators over 7 days. And aimed for elevated plasma, so slightly elevated plasma levels there. And afterwards, we looked at the lung function, histology, we looked at the T cells and the RNA-seq of the lungs. And it was kind of disappointing in a way because we did not see a detrimental effect on the lung function or the lung histology. However, I should caution that this was done only over 7 days and in vivo, so in humans basically, this progression from skin to lung takes years. So maybe this is just a time effect. However, what we found, and I found that very interesting, was that when we analyzed the lungs of the mice, we found the significant dysregulation of a few genes. And when I initially looked at the data, I was like, okay, that's disappointing. It doesn't really fit what we were hoping for. So that was a fail. But then I started looking deeper, and what we found was that those genes that are consistently dysregulated in the mice treated with those compounds, they are actually connected with the so-called circadian clock rhythm. And when you Google circadian clock and asthma, you will find that there is a growing body of evidence showing that circadian clock genes are dysregulated in allergic asthma. And that's exactly what we found in mice, because what it's also known is that the circadian clock genes are important to maintain the defense mechanisms, innate immunity, things like that barrier defense in human epithelia. And obviously the compounds that we use, they dysregulate. In some cases they upregulate, in some cases they abolish the expression of circadian clock genes. And it is well known that this dysregulation or this imbalance in the circadian clock genes can weaken or can cause pro-inflammatory effects, basically. And there is the one big question currently in the field is if this disbalance of the circadian clock genes might be one reason for the emergence or manifestation of things like allergic asthma. And so this is just a gene set enrichment study, basically, where you see what types of genes are upregulated, and then you can also see the pathway which is associated with that. And this is really very obvious, the circadian rhythm mechanism. And so basically, to sum that part up of my presentation, I mean, these are very initial findings, and there are many remaining or follow-up questions now. But what we think what's happening is really that in atopic dermatitis, there's an imbalance in the fibroblasts, and we don't really understand what causes this imbalance. But what we think to know is that these fibroblasts are somehow pro-inflammatorily active. And so what we think what's happening is because of a remodeling that occurs in the dermis, in the skin, certain pro-inflammatory factors are released into the bloodstream, and then they basically circulate within the human body. And at some point, something tips the scales in some people towards the manifestation of other allergic diseases, for example, in the lung. And so this is really something what we are trying to answer right now. What drives this priming of fibroblasts? Is it possible to use that for patient stratification or maybe also to predict the course of disease? Can we use that to prevent maybe this progression of inflammatory diseases? And what exactly do the circadian clock genes or the dysregulation of those mean? So this is something that that is basically work in progress now. With that, I would like to switch gears a little bit, and I would like to present a little bit of work that we have been doing in the area of next generation therapies. And so my lab embarked on that journey about 3 years ago now. And what I mean by that is that we started working on developing a gene therapy for severe genetic diseases of human epithelia. And because of the time constraint, I probably only focus on the skin work that we've been doing, but I'm pretty sure that each of you is aware of the advances and that we are currently seeing in genetic engineering or gene editing in general, which is mainly driven by CRISPR approaches or CRISPR-based approaches. And this is actually a press release that came out about 3 months ago. And this is the first data set that became available from the very first gene editing treatment that has been applied in the clinics. And what they did here was they treated people with blood disorders like severe sickle cell anemia and beta thalassemia. And what they did, they performed a one-shot gene editing approach based on CRISPR in those patients. And this is basically the results of that. And the data are fantastic. So basically all of the people with severe sickle cell anemia went without a crisis after this one-shot gene editing approach. And the same holds true for most of the people with beta thalassemia. Only 42 out of 44 required a blood transfusion. And this is huge. This is really unprecedented. And in fact, so the first approval for this CRISPR-based gene editing strategy is expected now within the next few months based on those data that I'm showing you here. And so my lab is not working on blood disorders, but as I mentioned at the beginning, we are interested in epithelia. And so we are basically trying to develop treatment options for severe genetic diseases of the epithelia. And I just put those here just to highlight the barriers that we are dealing with. So on the one hand side, we have the skin, which is basically ensuring our survival because it has a very robust barrier and it prevents the skin absorption of most of the environmental compounds and bacteria and radiation. And on the other hand, but again, I'm not sure if I will have the time to talk about that, there's the lung epithelium, which is also a first-line defense barrier, but it has a completely different barrier, which is the mucus here on top of that. So I'll start with the skin and the work that we've been doing there. And so these are just some exemplary images of patients with severe genetic skin diseases. And those severe genetic skin diseases are called genodermatoses. And one of those diseases that we are interested in is congenital ichthyosis. And genodermatoses in general are so-called orphan diseases, which means that these are very rare diseases. But as you can easily see from those images, these are really highly stigmatizing diseases. In some cases, they are life-threatening. For example, this one here, this is an 8-year-old boy suffering from epidermolysis bullosa. And this is a disease where the skin peels off. So it forms blisters and it eventually peels off. And so you cannot survive without skin. And so this is really a life-threatening condition. And the problem with those diseases is that there are no effective treatment options available and obviously no cure. But what I would like to highlight is that now, with the advances in gene editing and CRISPR-Cas, we finally do have the tools and the methods to really make a difference and to help those people. And that's exactly what we are trying to do. And so there are different ways to approach that. And maybe just to make the link back to the bioengineering and the tissue models, you may be wondering why I'm now switching to genetic diseases. Also for those diseases, no animal models do exist. The problem here is that although we know the mutation that caused this disease, and we can definitely knock it out in animals, but those animals with this knockout, they die immediately after being born because they have such a massive disruption of the skin barrier. So they have a massive transepidermal water loss, so they basically dehydrate. So there's no suitable model that we available in order to study this disease and to develop new treatment options. And so also for this project, we heavily rely on either excised human tissue or on bioengineered disease models. And I will come to that in a few minutes. So basically, there are two ways to go about that. One is the ex vivo strategy, and this is actually something that has been done already for this little boy. So what they did here was they took a biopsy from an unaffected area of the skin. They isolated the diseased cells, and then ex vivo they corrected the gene, or not really they corrected the gene, they introduced a copy of a functional gene in that regard. And then what they did is they grew large transgenic skin sheets with those corrected cells, and then they recrafted those skin sheets onto the body of this little boy. And the outcome was amazing. So I don't really know how this boy is doing now, but this publication was published in 2017. And back then, I think it was a 2-year follow-up. And the results were great in the sense that basically he survived, because that was really a life-threatening condition in that regard. And that the recrafted skin sheets, they nicely integrated with the rest of the human body, and it healed very well. So that is one way to go about it, but this has certain logistic challenges, is associated with logistic challenges, as you can obviously appreciate. So what we are trying to do is that part. We are trying to do that in situ. So basically the vision is to develop a topically applicable cream formulation, if you want, that we can just simply apply onto affected areas of the human skin, and which will then correct the disease-causing mutation in situ, so in the skin of this living person. And my lab focuses really on the non-viral delivery approach, because this is much, much cheaper. I mean, there are many approaches with viruses, but the problem is that most of those viruses cannot be packaged well with gene editing tools like CRISPR, because they are pretty big. And the other thing is that upscaling of those viral delivery systems is incredibly expensive, and cost is really an important factor here. With the non-viral delivery systems, and in our case, we are working with lipid nanoparticles, they are super easy to upscale and very cheap to produce. So there's a clear advantage of that. But what I would like to emphasize is also that just simply loading the gene editing tools into those particles and just applying that onto the skin will not do the trick. So what we need to do, even in diseased skin, is we need to physically modulate the barrier. Because even if it's very diseased, and it's clearly impaired, the particles would still be too big in order to really enter the skin and to reach the cells of interest, which are in our case the basal stem cells. Because when we would be able to correct the skin stem cells, that would really yield the most durable effects. So I already mentioned that we are working with lipid nanoparticles here. So the lipid nanoparticles are a mixture of helper lipids, cholesterol, ionizable lipids, and PEG lipids. And I'm pretty sure all of you have heard about lipid nanoparticles, because they have been a key ingredient of the current COVID-19 mRNA vaccines. And so the great benefit of those LNPs is that they are already clinically approved. We know that they are safe. We know that they can efficiently encapsulate genetic cargo. And so this is really a very good approach. And what we did was we started off by comparing different cargos. So we work with the Cas9, which is basically an enzyme which cuts the DNA. And this, we delivered that either as a mRNA or as the protein. And the reason why we bothered working with the protein was that there are some studies that show that when working with the protein, there are less off-target effects, because that's a major concern, that you edit some gene sequences that you don't want to edit, and that this may cause some long-term effects. And one of the biggest fears is basically that this would initiate tumor growth. And so that's why, for those safety aspects, we included the protein. And so then we did, over the past years, we did a wide a range of screening purposes, and we tested many different lipid nanoparticle formulations. And what you can see here is we compared the mRNA versus the RNP, and I think overall what we can conclude is that we see significant differences depending on the LNP formulation. And the other important takeaway message is really that we get editing rates between 15 to 20 or between 10 to 20%. And you may see this red dashed line here, and this is a very important line because this also is our benchmark. And I think I didn't mention that before, but when aiming to treat genetic diseases like the ones that we are targeting, already correcting a quite low percentage of cells would suffice to alleviate the most severe symptoms. So in case of those genodermatoses, it is anticipated that if we can only correct 10% of the disease-causing mutations, or 10% of the cells, that this would suffice in order to really improve the phenotype and the quality of life for the patients. And that's why our benchmark is really this 10%. And as you can see here, that we can easily edit more than 10% of this threshold that we set here. We also tried to further increase those editing efficacies by combining them with some additives. And those additives would be, for example, endosomal escape enhancers. I'm not sure if you are aware of that, but whenever you develop substances into a cell, when they are taken up via endocytosis, they basically end up in endosomes. And in order to really make a difference, what they have to do is to escape from the endosomes. And so for genetic cargo like CRISPR, it is there are data showing that only 2%, and let that sink in, so only 2% of what you deliver eventually escapes the endosomes. So only the 2% makes the efficacy or the editing, or is available for editing. And so what we are trying to do by using some endosomal escape enhancers is to bump this up to higher values, because then we could increase the efficacy. And so the data that I show you here, this is with the endosomal escape enhancers, and you can see that by combining that with the LNPs loaded with mRNA, we see an increase, although it was not the significant increase that we hoped for. So, but this is really at the beginning, and we are hoping by tweaking and modulating the approach a little bit that we can further increase that. It was a modest increase, but not the significant increase that we hoped for. So what we then did was we further screened a wide range of lipid components, and we really looked at different ionizable lipids, at the pKa of the lipids, at the lipid ratios, at the PEC concentration, and things like that. And this is really some very recent data where you can see that we screened, again, different lipid nanoparticle formulation, and there was one that really stood out, which is LNPH. And with this LNPH, it's quite interesting because all of a sudden we get 70% editing. And so we are very excited about that, because this is really the first time that we see this much of editing. However, I should note that we also saw quite some toxicity with this LNPH. Usually with the others that yield editing around 20%, we don't see that. And currently we are trying to modify this lipid here, this ionizable lipid that obviously yields this high editing rate, in order to improve the tolerability while retaining the high editing efficacy. But this is work in progress, so I don't really know if that will work out or not. So this is in 2D, but then eventually you have to translate that to 3D, because we want to do that in skin, right? And skin is a stratified epithelium, so we have different layers and not only a monolayer as we have it in cell culture. And so what we do is we use two different models. Either we get freshly excised human skin from plastic surgeons. So we directly collect it after the surgery. We transport it into our lab. It's still warm when it gets there. And then we basically physically pre-treat the skin. And then we apply our formulations. And this is just an approach that we did with a laser method that we are currently testing, and this is just to visualize the holes that we are poking. And what you see here above this yellow line, this is really the epithelium. Below the yellow line, this is the dermis, but we don't want to target the dermis. We really want to stay in the epithelium. And you can see that with the physical modulation approach that we are pursuing, that we can really create nice holes in the epidermis. And then here we applied lipid nanoparticles that were fluorescently labeled. And here you can see that the lipid nanoparticles really penetrate into the epidermal layers and give a nice red fluorescent signal. And then we also wanted to quantify that. So what we want to know is what is the actual editing efficacy in actual skin tissue. And so I think those results are quite encouraging, because we also, depending on how long you incubate that, we get editing around 10%. And this is, here we are really at the beginning, so we are very hopeful that by further optimizing this approach, we will be able to further increase the editing rates here. So we are very close, in some cases even above the threshold that we set ourselves. And now thinking back about this very efficient LNPH that I showed you, this was editing efficacy in 2D. And now you may be wondering, okay, what does it look like in actual skin tissue? So is that translatable? And so this is again work in progress. So I don't have a definite answer yet, but I can show you initial results that we have. And I find that quite interesting, because what we see from initial studies is that it does not directly translate. So we see again editing of about 10%. But looking at this high editing rate in 2D, we were hoping that we would see much more editing efficacy in actual skin tissue, but somehow this gets lost. And we don't really understand why, but it probably points towards that the physical barrier is more important in that case and kind of hampers efficient editing, but that's something that we are looking into at the moment. And what we also started doing, and this is really, I don't really have results at that point, but I just want to tell you the principle. So over the past years, we have collected patient cells from people suffering from those genodermatoses, because as I mentioned, there are no suitable animal models available. And with those patient cells, we grow disease models, so skin disease models, and they look like that. So this is a bioengineered skin model. And this is some older data where we have a skin model, a normal skin model. We see that this very specific enzyme is there. In the patient model, because of the mutation, we don't see this enzyme. And then here, in that case, that's from an older study where we substituted the protein. We were able to show that we were able to reconstitute the enzyme activity. And that's basically the model that we are working with. And currently, we are depending on the mutation that is present in those patient cells. We basically have to design a very specific gene editing strategy. And we are closely working with some geneticists on that. And in most cases, we can use some so-called base editors, which is a novel technology based on the CRISPR-based editing, but this doesn't really cause double-strand breaks. It's just, it exchanges the wrong base for the correct base pair. And so that's basically something that we are currently doing. You may also remember that I mentioned that initially we included the protein, the Cas9 protein, because we hoped that we would see less off-target effects. And this idea that a protein causes less off-target effect basically stems from this very publication in 2014. And what they showed very nicely is that when they delivered the Cas9 protein, that this was basically gone after 24 hours, and that this was not the case when you deliver plasmids or mRNA. And the longer you have the Cas9 in the cell, the higher is the risk for off-target effects. And so the interesting thing, what we found is that in our cells, and these are all primary epithelial cells, we can still detect the RNP after 72 hours. And what I would like to note is that obviously, the intracellular half-life of the protein is cell type specific. And here in that publication, they used a cell line, which really propagates very rapidly. And so it makes sense that here it's gone after a short while, whereas in epithelial cells, which have a rather slow turnover, it's still present after 72 hours. And so basically, to come back to this initial notion that RNP or the protein might be beneficial, currently we are thinking to let that go, because it's really difficult to formulate the protein in lipid nanoparticles. And obviously, we don't see this shorter expression or this shorter presence. And so we don't really see the benefit of pursuing that any further. And with that, I'm already talking 45 minutes, and that's why I will skip through the last part where I wanted to talk a little bit about the lung. And I'm just going directly to my acknowledgement section. I mean, obviously there were many people that contributed to that work and that actually ran the experiments in the labs. I would like to thank my lab at UBC, the collaborators at UBC, Peter Cullis, who's working with us on the lipid nanoparticles, and then companies like Nanovation and Acutas Therapeutics also working with us on the lipid nanoparticle formulation. And I would like to thank you for your kind attention, and I'm happy to answer any question. Thank you.
Thank you, Sarah. Really amazing results you show us. Let me put my camera again. Well, we have a little delay between our talk here and the transmission to YouTube. First, I'll have a doubt. For the cultivation of the fibroblasts and the cells from the skin, the matrix you use to cultivate them could change the results of the secretome or the genes?
Yes, that's possible. What I would like to mention there is that we, especially for the proteomics data that I showed you, and for the scanning electron microscopy data, what we did there was the fibroblasts produced their own extracellular matrix. So we did not put them into any collagen gel or things like that. So it was really native ECM. And so I agree, in the normal skin models, that could be the case, because depending on the environment, but when they produce their own extracellular matrix, there are no exogenous components to that. Yeah.
Yes, I'll ask you because the collagen is the most used matrix to cultivate cells, but today there is a lot of commercial matrices like Matrigel and other ones with different brands and names, and possibly, I guess, this could influence the culture.
Oh, it definitely does. I mean, this whole Matrigel story, this is really a painful story as well. As you know, I mean, it's a biological product, so there's a lot of batch-to-batch variation, and we experienced the same with collagen. And there are some initiatives to basically replace Matrigel or those collagens, either by letting the fibroblasts produce their own extracellular matrix, but this really takes a long time. So this basically takes additional 4 weeks. So the skin models usually are cultivated within 2 weeks. When we let the fibroblasts produce their own extracellular matrix, it's additional 3 to 4 weeks, because it just takes some time until they have produced enough. And so there are some chemists that we are working with, and they are worldwide. There are people that are trying to chemically or to synthesize alternatives to those Matrigels, and to replace it eventually in order to decrease the batch-to-batch variability and things like that. But I think really we're just at the beginning there, and this is an unsolved problem. I totally agree with you. Yeah.
Yes, I agree. Another doubt: when you use these nano systems to deliver the genes, how long do you know how long it takes to see a clinical improvement?
You mean in vivo?
Yes, in vivo.
That's a good question. I don't have a good answer to that. I mean, what I can tell you is that the skin turnover is 4 weeks approximately. So I would suspect it takes at least 2 to 4 weeks until you see something. Yeah.
Okay. We have questions here in the chat. Paulo is asking you, what's the new future possibilities and approach of your research?
Which one? The gene editing approach or the...
Which one, Paulo? The gene editing with the skin? I guess it is.
Okay, so yeah, what we are currently trying to do is we are looking for investors. So the plan is really to spin off a company and to develop that further. So we are, because I mean, this is a unique opportunity with this gene editing, and the patients are suffering so much, and there's literally almost nothing that they can do. And so I think there's definitely a huge need for that. And so we are trying to commercialize that. And the thing is that as long as you're in an academic setting, so in the university, the options are limited, you know, and as soon as you're in the company, there are more private investors. And I mean, this is a very expensive endeavor. So you need quite a bit of money to get that up and running. Our hope is that because those genodermatoses are rare diseases and there are special fast track options for rare diseases, so it's basically easier and less expensive to run the clinical trials to get approval and market authorization. And so we are planning to apply for those streams, which makes it a bit easier. But really the translation from bench to bedside, this is something that we are currently focusing on.
I found really amazing that 10% of the gene editing could improve so much the progression of the disease and improve the condition of the patient.
Yeah, and actually maybe 10% is even too much, because I know from a colleague of mine, he's working in a field that focuses on hyperlipidemia, like very high cholesterol concentrations and hyperlipidemic diseases. And what they are saying that in their case, already correcting only 2% of the disease-causing mutation would be a game changer. So that's quite encouraging. Yeah.
Yes. Because, well, change 2%, it's a number much more easier to obtain than maybe 50-60%.
Agreed.
Okay, there are a lot of people thanking. Thank you for the presentation. Finding that your results were very interesting and quite new and quite impressive. I don't know if we have any more questions here. And since we are right on time, I'll appreciate again, Sarah, your presentation. Thank you very much. I guess we can end it here. Mm-hmm. So have a nice end of the day.
Thank you very much.
Bom resto de dia, pessoal. E lembrando a vocês que na semana que vem o nosso seminário será presencial no anfiteatro da faculdade às 17 horas. Depois vocês vão receber os comunicados por e-mail e pelo Instagram do PPGCF. So, thank you, Sarah. Thank you, everyone. I will close the transmission.
Okay, bye. Take care.
Bye. Thank you.